The next topic I want to cover today is called nondisjunction, and this will help to appreciate the details that we went into looking at what causes hybrid sterility in mules, because that involved unpaired chromosomes. This is a way that we can have extra chromosomes in a cell even without being hybrids. So we've looked at a variety of different kinds of mutations. There were different kinds of point mutations where you had substitutions, you had silent substitutions, frame shifts, all those sorts of things. And then we saw uneven crossovers where you get gene duplication. The third major category of mutation is called nondisjunction. And this is an error that happens in meiosis. What we want to look at is that at the beginning of meiosis, we have our chromosomes set up with the sister chromatids lining up to form a tetrad. And then what should ordinarily happen through meiosis is eventually the tetrad gets split into one chromosome in each of 4 different germ cells. But with nondisjunction, the tetrad doesn't break up properly, as in this case here, or the sister chromatid doesn't split up. Either way, by the time you get down to the gametes, there's a few with an extra chromosome in it. Because this didn't separate, both members of the sister chromatid end up in the same sperm or egg. And likewise over here, there'll be extra chromosomes in these sperm or eggs. Now, this can cause real problems. And one of the most widely appreciated of these genetic disorders is called Down syndrome, which involves severe mental retardation, shortened life expectancy, and sterility. And there are certain rather conspicuous facial characters of children who have Down syndrome. But what's happened with this is that whereas most of the sister chromatids, the tetrads, they divided properly at meiosis, one didn't. And so if you have an extra copy of chromosome 21 because of nondisjunction, either with that tetrad or the sister chromatids, you get an extra copy of chromosome 21, and now everything's out of balance. So, our phenotypes are like the outcome of a very complex recipe with all those different genes acting in the right proportions. But with an extra chromosome, there's a whole bunch of extra protein being produced, and that throws the whole recipe out of balance. Chromosome 21 is associated with Down syndrome, and that's relatively a mild form of trisomy, having an extra chromosome. And it's mild because that's a relatively small chromosome with only a few genes being coded, and so extra proteins being produced at only a few loci. Another form of trisomy that is found in some children involves trisomy at the 18th chromosome. And this child was the daughter of a recent presidential candidate from 2012, Rick Santorum. And so she had trisomy 18. She had a third chromosome number 18. So again, the recipe's imbalanced. And as in the case of Down syndrome, these children with trisomy 18 have severe physical handicaps. Now, why is it that we see nondisjunction? And in humans with Down syndrome, there's a very clear relationship with the mother's age. If you look at the risk of a child being born with that extra 21 chromosome, chromosome number 21, it's very rare in young women. But as women get older, as they reach their mid-30s, by the time they're 40 and then through their 40s, chances go up quite dramatically. And a famous example of this, again, with a presidential candidate, this is the vice presidential candidate in 2008, the former governor of Alaska, Sarah Palin, and she had a child with Down syndrome. Now, what happens with this is because meiosis is not separating each one of those sister chromatids out of the tetrad into 4 discrete germ cells. And the reason there's an effect of mother's age is that every woman is born with all of her oocytes. And all the oocytes started the first phase of meiosis, even in the fetal stage of development. And so these oocytes are waiting and waiting and waiting in the ovaries until they're released at ovulation, and then at ovulation, meiosis resumes. After that very, very long wait, it just seems that these aging oocytes are less likely to show the proper distribution of each chromosome from the tetrads into the sister chromatids into the haplotype of the eggs. And so with Sarah Palin, she was 44 when she gave birth to her child with Down syndrome. So it's a definite problem in older women. Now, the reason for going into this in detail here is that having these extra chromosomes, which is ordinarily a severe problem, has a surprising consequence in the patterns of speciation in a lot of organisms on Earth today, in plants. And what we're now going to look at is a really extraordinary event, very rare in nature, where we have what's called complete nondisjunction. Complete nondisjunction. So we're going to have our diploid cell, and at the beginning of meiosis, everything lines up, but then everything shows nondisjunction. And so instead of producing haploid gametes, these plants produce diploid gametes. So you have a diploid parent producing diploid gametes. Now, if the diploid gamete were to be fertilized by a haploid sperm, or if a diploid sperm were to fertilize a haploid egg, the resultant zygote would be triploid. And these triploids then would have an extra chromosome all the way along, which would mean that at meiosis in their subsequent development, you'd have the unpaired chromosomes like we saw in the mule, so meiosis couldn't proceed. Triploids, therefore, are sterile. Now, here's the odd thing about plants. As I've shown a few times before, plants, unlike almost anything we're familiar with, like dogs and cats and people, plants produce both eggs and sperm in the same individual. And so plants can fertilize themselves, okay? Now, if we have this very rare event of complete nondisjunction, so a diploid organism now is producing diploid gametes, it might be producing both diploid sperm, diploid pollen, and diploid oocytes or ovules or eggs, okay? Now, if it then self-fertilizes, okay, this will produce what's called a polyploid offspring. So instead of having two homologous partners, okay, two copies of chromosome 1, one from mom and dad, it'll now have 4, okay, because it never went through that haploid phase. So now it's having 4 copies of everything. Now, these are very, very special in the sense that these can now go from the tetraploid state, and every chromosome will have a partner, so we don't have the problem of a mule, and they can produce, again, diploid gametes, and if they can find other diploid gametes to fertilize or be fertilized by, their children will then be tetraploid or polyploid again, okay? But they can only do this with a very, very restricted number of individuals, okay? So if you have a mutant plant, it can self-fertilize, okay? Because it is producing both diploid eggs and sperm, producing the viable tetraploid offspring, and then the tetraploid is producing diploid gametes again, and it can then either mate with itself or its mother, okay? So within that very, very, very small group of partners, its offspring will be viable, okay? It will work. And so what we have then in plants, very occasionally, is the production of a polyploid which can produce viable offspring if it mates with itself or its sibling or somebody within its close family, but nobody else. And the key point here is that they're now completely reproductively isolated from all the other plants of what was formerly the same species in the past. So here's a trillium, and this trillium is a polyploid. It descends from a diploid plant, okay? And with that very first strange, non-disjunctive trillium in the world could fertilize with itself. It's completely reproductively isolated from everywhere else. So henceforth now, evolution can proceed separately in that new form, the tetraploid trillium. Now, it turns out that this kind of polyploidy has been very important in the speciation of plants in general. 70% of all flowering plant species have a polyploid ancestry. So sometime in their past, some mutant individual developed those diploid gametes, then self-fertilized, and the subsequent species went from a diploid ancestry then to a polyploid lineage. And they could only mate with each other, they became reproductively isolated, and eventually became properly recognized, distinct, different species. So this is something that you can actually do in the lab. There are some botanists, plant scientists, in breeding plants deliberately will play around with polyploidy, and it's something that we know can happen, that every now and then you get this change from diploid to polyploid, and then you get the origins of a whole new species. In the deeper lineage of the plant taxon, you have the earliest plants were mosses and ferns. And again, through time, we see that at one point or another in the last few 100 million years or so, 95% of all moss and fern species have some sort of polyploid phase in their lineage. So they have polyploid ancestry. So this is actually a fascinating technique, it turns out, for plant scientists. Because as consumer of plant products, humans would like to eat convenient foods and not be annoyed by messy seeds, say in the middle of our watermelon. So if we have a polyploid or a tetraploid watermelon, and we cross it with a diploid watermelon, okay, the offspring will be a triploid. The triploid hybrid will then grow, it looks like a normal plant, but because meiosis fails, it can't grow seeds. So when you see things like seedless watermelons, those are most likely to be triploids, and plant breeders are deliberately taking advantage of the fact that when there's an odd number of chromosomes, there's that extra chromosome, like we saw in the mule, okay? But this, it's like all the way along, in every set of chromosomes in the watermelon, there's an unpaired partner, meiosis can't proceed, voila, seedless watermelon.